Alright, so we are going to answer Biology 1400, exam paper 2024. And our first question reads, the ester bond in a lipid is formed between 1. A hydroxyl group of glycerol and the hydrogen atom from the carboxylic group as a fatty acid. 2. Two hydroxyl groups of glycerol. 3. Two hydrocarbon chains of a fatty acid. 4. A methyl group of a fatty acid and the hydroxyl group of a glycerol. 5. An oxygen of glycerol and the hydroxyl group of a fatty acid. 6. I don't know. So for this question, I'm going to help with the pictorial image. Okay, so when we look at esters, okay, esters are formed when the hydroxyl group, sorry, just a minute. Okay, so when the hydroxyl group is okay, so you find this hydroxyl group of the glycerol combines with the hydrogen group found on the carboxyl group. Okay, that's what forms the ester. Okay, this is what we call esterification. So in this case, let's go back to our question that reads the ester bond. So that's why our question was, our answer, sorry, was A. Next question. Which one of the following statements is not a characteristic of homologous chromosomes? Okay, so 1, they have the same length. 2, they have the same centromere position. 3, they carry genes that control any given trait at the same location. 4, they always have the same type of alleles on the same loci. 5, they pair up during meiosis 1. 6, I don't know. So the correct answer here is actually 4. Okay, like you can see. So for instance, if this is your chromosome, right? Okay, we have what we call loci, where a specific allele is found. For instance, maybe an allele for green eyes, for instance. Okay, now this allele is not always found on the same loci of the other chromosome. Okay, it's not always the case. This person could have an allele for green eyes as small t as a recessive and may have inherited a big T from their parents. Okay, so they may not have the same type of alleles. Sometimes could be TT or dominant TT, so they could be quite different. Okay, we go to number 2. Which of the following is correct? 1, the positively charged partons of atoms move rapidly at nearly the speed of light. Positively charged parts of atoms are the smallest. Okay, this is wrong. Atoms don't move at the speed of light. Then 3, the negatively charged, okay, so they missed out. Okay, so 2 is the positively charged parts of the atoms are the smallest, which is wrong. Then 3, the negatively charged parts of the atom are heavy particles, which is also incorrect. 4, the negatively charged particles of the atoms travel around the orbitals, which is the correct answer. 5, the neutral parts of the atoms are the smallest. 6, I don't know. So what you need to know is that within an atom we have a nucleus, okay, which I'll label as N. This N has neutrons and protons. These make up the nucleus. Then around them we have what we call the electrons. Now these electrons are negatively charged. These are the ones that orbit around the that travel around the orbitals. Okay, next question. Enzymes and other proteins are synthesized in the, so the correct answer is the G1 phase. Okay, so it's within this G1 phase that several, and you know that the G1 phase is the longest phase that occurs. Okay, so this is where the enzymes and other proteins are actually synthesized. So this is as it is. Okay, we go to our next question. Identify the expected phenotypic ratio of a dihybrid test cross between heterozygous dominant, okay, so heterozygous dominant phenotype. So for instance, if you have this heterozygous dominant, okay, this is a lucky guess, this is just an example, and a recessive homozygous recessive, okay, for instance, it's like this. Okay, so when we cross these, for instance, HT, T, and HT, and you have dominant T and dominant T. So identify the expected phenotypic ratio of a dihybrid test cross between a heterozygous dominant phenotype and recessive homozygous phenotype. So when we cross this, of course it would have been quite a bit longer, so the end result is going to be 3 to 1 actually. Okay, so a dihybrid cross, actually we were supposed to cross it like this in this case. Okay, if it's dihybrid, for instance, it's like this, HH, and recessive TT. Okay, H, small h. Let's use small h right there. Okay. So for instance, when these two cross each other, okay, HH and H, small letter h, okay, and recessive, then this one is crossing, of course all the products are going to be the same. Okay, so what you notice here, you have, okay, so here you have a dominant, so the recessive, the dominant, you have 2, okay, 2 or 4, and this question was asking us to actually cross this and this. So what you find that in the end, it's actually supposed to give us 3 to 1. Okay. So on account of time, I'll make I'll explain this later on. All right. Seeing that we have 100 questions to go. Okay, the next question is 6. Identify the non-reducing sugar. So from these structures, what you need to know, or rather what you need to point out here, is that just look for sucrose, because sucrose is a non-reducing sugar. Okay, sucrose is made when glucose and fructose combine. Okay, so when you look at these structures, the only one with fructose and glucose is actually that's 2. Okay, 2 is the one with glucose. So let's erase this and make a correction. Okay, let's just erase this. Okay, so this is what makes glucose, and sucrose, a non-reducing sugar. So what you notice is that I'm going to explain more. So the bond that's here, so glucose has an aldehyde group. Okay, so the carboxyl group is on carbon number 1, while fructose has an has a ketose, meaning the carboxyl group is on the second carbon. Okay, now in this bond, it bonds in such a way that there is no room to actually form a chemical reaction. Okay, hence it's non-reducing. When you test for starch, you'll find that instead of forming that blue-black solution, it doesn't form that because it's non-reducing. Okay, so it just means that the chemical wasn't able to penetrate, hence it just remains the same color. Okay, next question that we'll look at, question 7. Okay, the atomic and mass numbers of neutral sodium are 11 and 23. Okay, so let's write it down. The atomic number and mass number. So the atomic number of sodium is 11. So let's write it like that. Sodium, this is the atomic number. Mass number is 23. So yes, so that's the, so they're asking what's the mass number. So the atomic and mass numbers of the neutral atom are this and this. Okay, so what's the mass number of sodium plus? Okay, so they're asking what's the atomic number of sodium plus. So sodium plus, this is to, let's do some mathematics. So what we notice is that the mass number, just a minute. So we have to ask our question, how exactly do we calculate mass number of an atom? Okay, so I'm going to go to the drawing board where we're going to write this down. So let's just erase this. Okay, so mass number, okay, refers to when you calculate the number of protons plus the number of neutrons. Okay, so the atomic number is also called the number of protons, right? So the mass number, for instance, if it's 23, right, it means there were 11 protons and how many? So for us to find how many neutrons, we say 23 minus 11, thereby giving us 12 number of neutrons. Okay, so we go back to our question. So all we needed to do is actually to find, so here they wanted the mass number. So here they said if it's neutral, what does this mean? Okay, so when they mean sodium plus, it means there's obviously an extra electron that's on this. Okay, so let's just make a correction there. Okay, so an electron has been added there, hence it's going to be the mass number will change from 23, is going to be 24. Okay, because we're counting, so because protons, all these have charges. Okay, let's go back to the drawing board. All these electrons are negatively charged, right? Electrons are negatively charged, and you also have, so within your nucleus you have your protons and your neutrons. Your neutrons being being found there, and of course these are electrons that are found around. Okay, so what do they mean when they say sodium plus? Okay, so what we also need to know is that when you look at the electrons and the protons, let's just go back here. So I need to explain something here. We will go to biomolecules. Okay, so we need to know the exact details. Okay, so when it comes to this is what's actually going to help us. Okay, so we look at here, I'm going to highlight electrons are negatively charged, and the smallest. These are the ones that are found in the shells or orbitals, and at the center we have an equal number of protons which are positively charged. Neutrons have no charge. Okay, so the mass number of an atom is determined by the number of protons and neutrons, like we mentioned. Okay, so I'm going to use this information to explain what we're talking about. Remember, protons are positively charged, electrons are negatively charged, while neutrons, these have no charge. Okay, so what we need to notice is that the neutron sodium had 11 protons and had a mass number of 23. So what happens when this what happens when it's now charged? Okay, what happens to the mass number when this is actually charged? So for instance, when it becomes sodium plus. Okay, so what you also need to notice is that the number of protons is equal to the number of electrons in overall. Okay, so positives and negatives, you know that these two cancel each other out. So what happens is that if this sodium lost if it lost an electron, it remains positively charged because those other protons and electrons have cancelled each other out. Okay, so this is what we are referring to. Let's just go back to our question. So basically what we are trying to say is that our mass number in this case, just a minute, let's erase this. All right, so you know that if it's lost an electron, then the mass number is actually expected to remain constant. The atomic number is expected to remain constant. Okay, so however, so the nearest answer that we can pick here is, let's just highlight it here. Okay, just a minute. Okay, so the mass number isn't supposed to change as such because this atom actually lost, if it lost an electron. Okay, so in this case we would go for 22. Okay, because here they're asking for the mass number, not the atomic number. All right. Okay, we go to question 8. Dash can induce changes in the electrical charge of the neighboring molecules. So here the answer is van der Waals forces. Why? Because these are weak and non-specific charges that are caused due to interaction. Okay, so for instance, if this is your molecule, this molecule is slightly let's say positive. The more it comes close to another molecule which is neutral, it has negative, positive, negative, it induces a charge such that these positive charges attract the negative charges. So if these are positive, it's going to induce a slightly negative charge on the neighboring molecule. These are van der Waals, and the closer they are together, the more powerful the forces are. So the more far apart, these forces are going to be weaker. Okay, next question. Okay, let's just erase that. Question 9. Which of the following molecules represents the unsaturated fatty acids? Okay, so what you need to know is that this actually has a formula. Okay, so the formula for unsaturated fatty acids is CnH2n minus O2. Okay, this is the formula of the unsaturated fatty acids that have one bond. So this is the only answer that was able to tally with the actual formula. Of course, they didn't make a mistake there. Okay. Next up, we are going to talk about going to question 10. Substitution is a type of gene mutation where a dash is replaced by a dash. So substitution is where you replace in a transverse substitution, it's where you replace a purine. It's why it's a type of gene mutation where a purine is replaced by a pyrimidine. You know that purines have 2 rings, pyrimidines have 1 ring. Okay, so what happens is that this purine is going to be replaced by a pyrimidine. So this is one of the types of mutations that are found. Number 11, which of the following are the strongest in aqueous medium? So the correct answer is covalent bonds. Okay, so covalent bonds are strongest because they are sharing electrons. For instance, water, right? Or rather carbon dioxide. Okay, for instance, is CO2. So carbon is sharing, you know that carbon has 4 bonds, right? Okay, so if this is carbon, so right here where these lines are, they're sharing electrons. This doesn't easily break off in water because they are sharing, unlike where it's a ionic bond where there is one is gaining, one is losing electrons. They can easily disassociate when they're in an aqueous solution, but in a covalent, because they are sharing electrons, they need each other to survive, so they can't easily disintegrate. That's why they are the strongest in the aqueous solution. Next up, bacteria in E. coli on N15 medium is transferred to N14 medium and allowed to grow for 1 generation. Okay, just 1 generation. DNA is extracted from this bacteria and is centrifuged. What density distribution are you to expect? So N15 and N14. So these are grown in 1 medium for 1 generation. So the correct answer is 1 hybrid of N14 and N15. So what happens is that the DNA from N15, during replication, they share. Okay, so for instance, there'll be 1 strand with N15 and the other strand will be N14. Okay, so this is what happens because remember DNA is semi-conservative. So there's 1 old template and 1 new template. So in the first generation, you're going to have 1 hybrid N14 and 1 hybrid N14 and N15. Okay, 12. A group of students, sorry, is this 12 or 13? Okay, 13. A group of students crossed pea plants that had round and yellow seeds in a dihybrid green seeds, 28 wrinkled yellow seeds and 9 wrinkled green seeds. Determine the chi-square for these results. Okay, so I'm going to need you to pay particular attention because we are going to do some math. So from that question, let's just make this board free. Okay, so the question is saying it's a dihybrid crossbreed. So already you know that the distribution is going to be 9 to 3 to 3 to 1. Okay, for the round yellow and the green wrinkled yellow and the wrinkled green seeds. So to calculate the total number of offspring, we need to add up the expected. We need to add up the expected number. So for instance, we have 28 round yellow plus 9 round green plus 28 plus 9, and our total is 74. Okay, so to calculate the expected total number of offspring, we're going to say for the round yellow, 9 out of, when you add the ratio, these are ratios, right? So when you add these all, they're supposed to give you, so 9, you have 10, 11, 9, 10, 11, 12, 13, 14, 15, 16. So 9 out of 16, what's the probability? Okay, out of these 74, how many are going to be round yellow? So this is what we're talking about. So it's going to give us 41.65. Then we have round green, which is 3 of 16 times 74, which is going to give us 13.875. Then we have wrinkled yellow. These are 3 out of 16 times 74, giving us 13.875. Okay, same ratio. Then we have wrinkled green now, 1 over 16 times 74, giving us 4.625. Okay, let's get another page. All right, let's see if we can get another page. Okay, so from those values, how to calculate the chi-square? This is the formula: x squared is equals to the summation of the observed minus the expected divided by the expected. Of course, this is to the power 2. So for the round yellow, we go back to our previous, we're using these values, okay? Okay, so we're going to say 28, which is the observed, okay, which were given in the question, minus the expected. Remember the expected, these are the ratios, okay? These are the ratios. This is what we expect. So 28 minus 41.625 divided by 41.625, okay, times to the power 2. So when you do all the calculations, it's going to give you 4.46. Then we go to the next. You say 9. We're going by ratios, remember. We say 9 minus, let's go back to the previous page, 9 minus, remember, the red green minus 13.875. 13.875 over 13.875. You square that, the total answer will give you 1.71. Okay, go to the next page. You have 28 minus, I'm using the same figures that we used that side, minus 875 over 13.875. okay, square that, it's going to give you 14.38. Then the last one is 9 minus 4.625. You square that over 4.625. Remember, I keep on using the same figures we used in the first place. Let me use a different one. So these are our expected, okay? Our observed are these. Okay, now we are here. These are the ratios. We're now here. This is our observed 9 minus the expected over the expected. This is what we find. Now remember that chi-square, it's a summation of all this. So in the end, what we do is that you add all those that we found, 4.46 plus 1.71 plus 14.38 plus 4.14. Okay, you're going to find 24.69. Now notice that 24.69 is options. So what you do, it could be that they wrongly matched, they made a mistake on the observed numbers or the expected ratios. So when you find your case, you choose an answer that's quite closest. Now for 14 we can't answer because the question wasn't clear. They might have not typed it. Okay, so I go to 15. What role do hydrogen bonds play in the properties of water? Okay, so 1, they decrease the boiling point of water. 2, they contribute to the high specific heat capacity of water. 3, they make water molecules less cohesive. 4, they reduce the melting point of ice. 5, they increase the adhesion between water molecules. 6, I don't know. So the correct answer is they contribute to the high specific heat capacity. Okay, because of these hydrogen bonds, you know, water is like this. So between oxygen and hydrogen, there's what we call hydrogen bonds. Okay, these hydrogen bonds are being formed because of that interaction. Now, because they're so closely, there's so many water molecules that are tightly connected to each other, it's very hard. The heat that is required to break those bonds is so much. That's why it's called high specific heat capacity. All right, because there's so much heat that you need in order to break those hydrogen bonds, are the ones that cause water to have a high specific heat capacity. 16. Choose the statement which is true about alleles. A or 1, alleles are the alternative forms of a gene. 2, genes are the alternative forms of an allele. 3, the different alleles of a gene are located on the same chromosome. 4, traits are alternative forms of alleles. 5, alleles are linked genes, alleles of linked genes are located on different chromosomes. 6, I don't know. So when you look at alleles, these are alternative forms of a gene. Okay, like I explained, when you look at a chromosome, okay, these are chromosomes. An allele is a specific characteristic of a gene found in a specific loci. Okay, so if this person has an allele for tallness, okay, but they inherited the other T from their mother maybe, which is recessive. So these are alternative forms of the gene. It's an alternative. It can either be tall or short. So the person is tall or short, being expressed, one from the mother and one from the father. Okay. Number 17, hydrogen bonds can form between 1, water molecules, 2, a nitrogen atom in a molecule and a hydrogen atom in another molecule, 3, hydroxyl groups in the carbon 2 of each glucose residue that forms amylose, 4, NH and carbon to oxygen group in a polypeptide backbone. 5 of the above. 6, I don't know. So the correct answer is all of the above. Hydrogen bonds are not per se just limited to hydrogen and oxygen. Hydrogen can also bond with oxygen on this bond. Okay, can also, so hydrogen, what we need to know, hydrogen bonds can be with oxygen, they can be with nitrogen, okay, and so on and so forth. So these are the bonds that occur. Then we go to our next question. Our next question reads, which of the following bonds is the weakest? Again, the answer is van der Waals forces. Okay, because they are weak and non-specific. The force is dependent on whether they are close or not. The closer they are, the stronger. The further apart they are from each other, the weaker. Okay, so because of this, they have what we call fluctuations, and that's why they are the weakest. Okay, next is flagellin. Flagellin is a projection on the cell surface. 2, an organelle in prokaryotic cells. 3, a protein found in the cell wall. 4, a glycoprotein found in the projection of the cell surface. 5, a protein found in a projection in the cell surface. 6, I don't know. So flagellin, for instance, is your bacteria, right? You have the tail here. So we have what we call flagella. Flagella is what helps the bacteria to be propelled forward, okay? Now, within this flagella is what we call a protein called flagellin, okay? So this flagella has what we call flagellin, or rather flagellin is what makes up the flagella or flagellum. 20, gene mutation in sickle cell disease is an example of 1, frame shift mutation, 2, silent mutation, 3, neutral mutation, 4, missense mutation. Okay, so here I'm just going to run you through some of the types of mutations that we have. So a silent mutation, I just want to highlight this so that you can just hear. So a silent or null mutation is when the amino acid that's coded for okay, so it's when the amino acid is coded. This regardless of whether there's a change or not, the amino acid will still be coded. So it's called a silent. It's silent because you can't even tell that the mutation occurred. A neutral, so a neutral one is where the protein will not change much because the amino acids coded for are functionally the same. Okay, so this one has to do with the end results. Okay, the protein that's coded for will not change because the amino acids that make up that protein they function in the same way even if they've been changed in the first place. So for our question, it's a missense mutation. Why is it a missense mutation? It's because when there's a change in that base, okay, for instance, maybe AUC, when you change cytosine to adenine, for instance, it's AUA, this changes the amino acid into a totally different thing, for instance. So as an example, so that's what happens in sickle cell, okay? In sickle cell diseased patients, this is what happens. So this is what happens where you have a different amino acids. For instance, GAA is hydrophilic glutamate, but then when it changes to GUA, because the mutation occurred at the second codon, it becomes hydrophobic. Hence, the sickle-shaped red blood cells, they curve inward, giving it this shape, okay? Because they are now hydrophobic.